Hello and welcome to Porous Media Tea Time Talks. This is a platform for PhDs, postdocs, and young researchers or early career researchers to present their research to the worldwide society of porous media and gain the traction that they need to advance their careers and also do the networking necessary for this stage of research and academic career. My name is Mohammed. I'm a researcher at the University of Oslo, and today with me I have Kira Riccardi from Politecnico Milano and Ramin Muqaddasi from Sweden. Ramin recently graduated from the academia and joined industry in Sweden. Without further ado, I want to introduce our first speaker, Silvia De Simone. Silvia is a Ramon y Cajal researcher at the IDAEA Institute at the Spanish National Research Council in Barcelona, Spain. She obtained her undergraduate and master's degrees in civil and environmental engineering at the University of Naples, Federico in Italy, and her PhD in geotechnical engineering at the Politecnical University of Catalonia in Spain. As a postdoctoral researcher, she was enrolled in the subsurface CO2 group at the Imperial College of London and the Institute of Geosciences in Rennes in France at the National Center for Scientific Research, CNRS. Her research focuses on quantitative hydrogeology and computational geomechanics with a special focus on coupled thermohydromechanical processes, THM, occurring in deep fractured and porous formations during development of geoenergy activities. Thank you, Silvia, for joining us today, and the floor is yours.
Thank you for the nice presentation, and thanks for the invitation. I will start my presentation. Please do stop me if there is any technical issue, please. So I guess you can see the screen. So this presentation is about a work performed in collaboration with Caroline Darcel and Philippe Davy from the Fractory Group in France. And also with Hossein Kassani from NWMO in Canada and Diego Masivars from SKB in Sweden. And the topic of the talk is about Biot and Skempton coefficients for fractured rocks. So let me first start by introducing these two coefficients that are very important for the hydromechanical behavior of geological materials. So when we are dealing with fluid-saturated porous materials, we know that the deformation that the material has in response to an applied stress is not proportional to the total applied stress, sigma, but it's proportional to the effective stresses. So this is different to what happened with dry materials, in which the deformation is just proportional to the applied load. So when we are dealing with porous fluid-filled materials, because of the presence of the fluid in the pores, the load is partially acting on the pores and therefore on the fluid pressure, and partly acting on the solid skeleton in the form of effective stresses. And these effective stresses are those that ultimately control the deformation of the porous saturated materials. And the balance between effective stress and total stress and pressure is given by this Biot coefficient alpha. This law applies in every moment whenever we are in drained or undrained conditions. But when we are in undrained conditions, and which means in short time for geological materials, there is also another important law. And this law applies when we apply a load. We know that there is a proportional increase of pressure, which depends on the Skempton coefficient B. So it is clear that when we are in undrained conditions, the product between alpha and B defines the effective stresses. And therefore, it gives us a measure of the potential deformations and also of the potential for failure for the material. So we are particularly interested in these two coefficients because this is relevant to a large number of applications that include both natural situations, like in the case in which the load changes because of glaciation or deglaciation, but also in human-induced situations, like, for example, in the case in which we have extraction or excavation or constructions. But also it applies in cases in which we are changing the volume of water or the volume of fluids in the subsurface, like in the case of volume extraction or injection. So talking a little bit more about these two coefficients, we know that they change between 0 and 1, and in general they are close to 1 for highly compressible materials, and they are smaller than 1 for stiff materials. And these coefficients are generally directly or indirectly measured in the lab, but they can also be calculated from known properties, from known mechanical properties through theoretical expressions, which are, however, valid for homogeneous isotropic materials. So by using lab experiments or these theoretical expressions, it has been shown that these coefficients largely depend on the pores and on their shapes. And in particular, for example, here for the case of Biot coefficient alpha, experimental results show that alpha increases with the porosity of the material, of the porous material, but more sophisticated analysis has also shown that it depends on the shape of the pores. So if the pores are more elongated, the Biot coefficient is larger than if the pores are spherical. And it also, it has been shown that the alpha is also anisotropic. So depending on the orientation of the load with respect to the shape, to the elongated pore or crack, the coefficient is larger or smaller. So these estimations are very important, but they are limited to the sample scale. And we wonder what happens when we are dealing with large-scale rocks that contain fractures. So this topic has been approached by several researchers in the last 20, 30 years. I'm not getting into the details, but here there are some references. Most of these approaches, however, are limited by some difficulties. So most of them are limited to D or to sample scale. And in many cases, there is no consideration about the fracture orientation nor the fracture volume. And the intact rock is considered porosity-free. So we want to take all these points into consideration in our analysis, and we want to define equivalent Biot and Skempton coefficients for a saturated fractured rock mass. So we did that by defining our coefficients with respect to an average stress, like is indicated here in these expressions. And we derive theoretical estimations for these two equivalent coefficients by applying the poroelasticity theory and a total volume deformation approach under an applied incremental stress and different hydraulic conditions. And we do that by assuming that our porous fractured rock is composed by a fractal network with many fractures inside, and by an intact rock that is porous and that is considered homogeneous. So the intact rock is homogeneous and isotropic, but then inside this rock we have a fractal network with many fractures. And we also assume that the total volume deformation is given by the sum of the deformation of each individual fracture and the deformation of the intact rock. So we arrive, by using this approach, we arrive to an estimation, to two theoretical expressions to estimate this equivalent coefficient alpha and B, in which, as you can see, essentially they can be evaluated by means of some known properties of the fractal network, like fracture size and aperture, fracture normal stiffness, and also the orientation of the fracture with respect to the initial stress and with respect to the applied stress. And also knowing, of course, some properties of the intact rock. And as you can see, these expressions are weighted averages of the Biot coefficient and Skempton coefficient of the respective components, so the ones of the intact rock and the ones of every single fracture. And the weight is this gamma, which is a parameter that measures the contribution of each component to the total deformation. Also, I would like to draw your attention to this parameter theta that appears in the equations, and this parameter depends on the orientation of the fractal of each fractal with respect to the applied stress tensor, which means that our equivalent coefficients, although they are defined, they expresses the variation of effective stresses and pressure with respect to the average total stress. They despite that, they depend on the specific stress tensor that we are applied. So they will not be the same if we apply two different stress tensor with the same average stress, but for example, if we apply stress tensor when the largest stress is in the horizontal direction or in the vertical direction. So these expressions are being validated against numerical simulation in 3DEC. But I'm not going to into these details. I'm just telling you that they work very well, and you must trust me in that. And because now I would like to discuss a little bit by using these expressions to discuss how these equivalent coefficients change with respect to the properties of the fractal network that may change broadly. So, so for now, let's keep something simple. So let's keep that fracture aperture and stiffness are the same in all the fractures, and we just change the density of the fractal network. So we increase the number of fractures in our network, and you see here that both alpha and B increases with an increasing fractal density. As you can see, they move from the value of intertrode to larger value. So we have to take into account the presence of the fractures when we are estimating alpha and B, because otherwise we are making an incorrect estimation of the hydromechanical behavior. Then, keeping all the same assumption for aperture and stiffness, we can also observe that these two coefficients change with the stiffness of the fractal network. So they both decrease with the increasing fractal stiffness, and they slightly depend on the aperture. In particular, B decreases with increasing fractal aperture, but Biot coefficient is not depending on the fractal aperture. So this means that we have some uncertainty in evaluating these coefficients because these properties are generally unknown when we deal with deep rocks. And finally, I want to show how much these coefficients depend on the orientation of the fracture. So here, when we move from the blue lines to the red lines, we are changing the orientation of a set of parallel fractures. So we move from horizontal fracture with respect to a vertical applied load towards vertical fractures, and you see that especially Skempton coefficient decreases. So we see that it is the Skempton coefficient is larger when the applied stress acts normal to the fractures, and in case of alpha, the opposite occurs, but the effect is quite limited. So the coefficients are quite anisotropic. So now let's break this assumption that we have done about aperture and stiffness, because actually in reality we know that fracture aperture and stiffness are depend on some properties, and in particular they depend on the stress. So what we know is that we have a fracture, and the fracture will have a depth and a certain orientation, and there will be a certain in situ stress acting on it. So the larger is the stress acting normal to the fracture, the smaller will be the aperture, and the larger will be the stiffness. So if we now take this into account, and we again look at the effect of the orientation, so we have some additional effects. So here in this slide, the orange line represents the results in which we do not take into account the effect of the orientation on the aperture and stiffness. We only consider the effect that we have already analyzed before. And so we see that if we move the fractures from horizontal to vertical, the product, now we are looking at the product, alpha B decreases. But now we see that depending on the in situ, so on the initial stress field, we can have this effect amplified, like in the green case here, or we can have this effect reduced, like in the blue case here. So we have two effects of the fracture orientation. And also, fracture aperture and stiffness depend also on the size of the fractures. So larger fractures are more compressible and more open, and that has been shown in the literature by many authors. And so if we acknowledge this effect, And we also acknowledge the fact that the distribution of the fracture size in rocks generally follows a power law. So we have a few large fractures and many small fractures. So we now combine this information all together, and we analyze what happens when we acknowledge this proportionality of aperture and stiffness with the size of the fracture, and we acknowledge the power law distribution. And now what we observe here, if we change the value of the exponent omega, so if we move from systems that are composed by a few large fractures to systems that are composed by many small fractures, and we are keeping the density of the fracture the same in these cases, we observe that alpha and B are larger if the system is composed by a few large fractures. So the mechanical behavior, the hydromechanical behavior is much affected by the organization of the fracture size, not only by the density. So to wrap up, we have seen that the fracture densities the fractures play an important role on these equivalent coefficients, and the larger is the density of the fracture, the larger are the two coefficients. Then we have seen that they are highly anisotropic, and they both depend on the orientation of the applied load, but also on the orientation of the initial stress and on the fracture orientations. And then we have seen that there is an impact of the distribution of the fracture size, and so the coefficients are larger when the systems are populated by a few large fractures. So in the end, the fracture contribution is larger in systems that contain large fractures that are oriented parallel to the largest principal initial stress and normal to the applied stress. So although this is a kind of recipe, we know that there are a broad range of uncertainties because some of these parameters are quite difficult to be constrained with in situ measurements. So here there is a list of references in case someone is interested in more details. And with that, I conclude, and thank you very much for your attention.
Thank you very much, Silvia, for this very interesting talk. It was really interesting to listen to and hear about your work. It was also very interesting to hear about your work in a way that you use this approach from, breaking down the, problem into the smallest pieces and then, building up the whole model. So I ask the dear audiences to put their questions in the chat so we can take them. But at the same time, yes, we have two talks. Yes, so we have one question or comment from, I think, Professor Hassanizadeh. Very nice talk. You introduced the coefficient alpha as a scalar and in a scalar equation, but then you mentioned that is anisotropic. So if you can just comment on that.
Yeah, that's a very good question. So I introduced the coefficients as a scalar because, so first, let's say that the coefficients can be defined in the literature as scalars, but also as tensors. In this approach, we define the coefficient as scalars because we define the coefficients with respect to the average stress, to the average applied stress. But they are anisotropic because they do depend on the applied stress tensor. So the point is that we do not define the coefficients we depend to the applied tensor. We define them in the equations with respect to the average stress. So essentially, what we say is, for example, for the Skempton coefficient, we say the increase of overpressure is equal to our equivalent Skempton coefficient multiplied by the average stress, the applied average stress. But the coefficient itself depends on the specific stress tensor that we are applying. So in these terms, the coefficients are anisotropic. I hope that I replied to the question.
Yes, I will check the chat if there's any more update on this question. But I also have a question of myself, because I was not really able to follow if you did any kind of simulations with time-dependent resolution like, do you also change the time if you want to basically do a simulation, or is it a constant time and then you just change other properties to see what the effects are?
Yeah, so this kind of problem is a kind of static problem, because the so Skempton coefficient is defined in undrained conditions. So essentially there is no fluid flow, or the fluid is there in the pores. You apply the stress, and therefore you have a instantaneous increase of pressure, and you don't allow the flow. You don't allow any flow, so you don't have any transient behavior. Everything is static. And then for the Biot coefficient, so this is something that apply both in drained and undrained conditions. So to evaluate the coefficient, what we did is to assume that we have a known pressure in our system, and like an imposed one, imposed pressure and an imposed total stress, and we derive the expression for the alpha in this way. So this was done both in the theory and in the numerical simulation. So there is no transient behavior. Everything was static essentially. So we applied in the case of the evaluation of the Biot coefficient, we applied an imposed pressure in the system everywhere.
Mm-hmm. Yes, thank you so much. Just checking if you have any other questions. Otherwise,
I also have a question, if I can.
Yes.
Yeah. So I'm totally ignorant in this topic, so sorry if my question is maybe stupid, but I was wondering, you were mentioning at the end that it's really difficult to have like an estimate from an experimental point of view of the parameters of the fractures and of the rock. And I was wondering if you can comment on that. So like, how do you estimate those parameters generally?
Yeah. So I didn't have the time to also show these other results. So we analyzed like ranges of reasonable values for stiffness, for fracture stiffness, and for fracture aperture, and essentially there are some. So there is some uncertainty, but you can kind of reduce it if you consider ranges of values. The point is that it depends. So in some conditions, and also depending on because everything is really kind of weighted averages. So in some situations, the fractures they have an important weight, and in some other situation they have less weight. So essentially, since this gamma factor depends on the stiffness and of the fracture and is inversely proportional to the stiffness, so essentially it is fractures are more important if they are more compressible. And so we can have a kind of change of values depending on this. So we have analyzed ranges of values, and the idea is that with some knowledge about your rock, you can kind of constrain the ranges. But of course, you will always end up with a certain uncertainty.
Okay, thank you. Thank you so much.
But I think it's good to know that it's good to know that these parameters can change in any analysis that is done, because very often these two coefficients are assumed equal to 1 for simplicity, and what is shown here is that they can be much smaller than 1, and they can broadly change with some parameters. So it's important to take this into account.
Sure. Thank you.
Thanks.
Yes, thank you so much once again, Silvia, for joining us today. It was very interesting to listen to you. And we move on to our next speaker. Aurelia Bucciarelli from the University of Bern. Yes, sorry for that.
No worries.
She's a fourth-year PhD student at ARTORG Center at the University of Bern, and she's within the cardiovascular engineering group. She has a background in mechanical engineering with a focus on biomedical engineering. She does research on dynamics of red blood cells in capillary networks, and she does experiments with real human RBCs, or red blood cells, in microfluidic chip, which feature channels of dimensions of 8 micrometers size. And within the research group where she works, they do research on RBC dynamics at capillary bifurcations and distribution and velocity of RBCs across the entire network to examine the responses to changing channel dimensions, simulating parasite activity. So without further ado, floor is yours. Yes.
Yeah, thank you very much. So yeah, you already introduced a little bit, but I will start generally with the brain, because brain is really important for all of us to do our research. And it's actually accounting only 2% of the body weight, but to function, it needs 25% of the body energy. And it doesn't have any storage in it, so it needs a constant supply of it. And this energy is mostly oxygen and glucose, which is transported to the blood, which is therefore really important. And from the cardiac output, so every time that your heart is beating, 50% of the blood that comes out of the heart needs to go through the brain. Otherwise, we will not be able to work and do all the beautiful stuff that we can do. So it's really important to understand how blood flows through the brain and how does it work, essentially. And if we look, wait, I can put the pointer. So this is actually a slice of a mouse brain, but a human brain the structure is really similar, where you have the major vessels on top of the brain. So you have in red here the arterioles and in blue the veins, which bring the blood to the brain and then back to get oxygenated. And then you have some descending arterioles and ascending veins, which bring the blood inside the brain. And then in between those, you see this, here is in gray, here there are some green parts. This is the capillary network, and is where the oxygen exchange happens, and which is very important. The interesting thing is, for the arterioles and the veins, you have a more tree-like structure. So if from going to A to B, there is only one way. There are not multiple ways to get there. But the capillary network is a network, and therefore to get from one point to the other, you could choose multiple pathways. And this is what in our lab, or in my big project in the lab, is concentrating about, understanding how red blood cells flow through the capillary network. To do so, we are not working in vivo, and we have now a small group that does also simulation, but I mostly work experimentally. So we have different kinds of microfluidic chips. These are really easy and just one channel, so not a network, but we also work with more complex, obviously idealized network, as you can see here. And the chips this is one chip that I produce. So they are really, really small. And the channels are, I said between 8 and 10 micrometers, which is just a little bit bigger of the size of the red blood cells. And what we look at generally in our research is more generally, how do red blood cells divide or spread throughout the network. This is an example from a previous publication, where you can see in this network we have some channels. Here there is a channel, but there is no red blood cells, because actually the distribution of red blood cells in the capillary network is heterogeneous, it's not homogeneous. And then, well, you mentioned it. We also check or look at what are the influences if we change some dynamic properties of the network. So in this case, we change the size of each channel and analyze, how now do the red blood cells distribute? Do they distribute differently? Do they go faster or slower? What are the influences of the small changes? And this is more the red blood cells dynamic at the network level. And I would love to give you some new results on this, but unfortunately the last experiments are still being analyzed by my computer, so I could not give you new results on this. But what we also do with the simpler model that I showed before is we wanted to look at just one bifurcation and not concentrating on the whole picture, but just concentrating on one bifurcation to see what are there maybe some causes at this intersection that lead to this heterogeneous distribution. So I will speak mostly about this. But before going into this, I need to introduce one concept, which I only heard about it in the capillary system, but you can think about the capillary system as a porous network, because you have some materials where around it flows fluid, in this case blood. So it's two-phase fluid. So I don't know if this also happens maybe in the ground with some bacteria, but it's really interesting that if you look at, so if you have a bifurcation, this is a bifurcation where you have the parent vessel and two daughter vessels. If the vessels are really big, you have a distribution like the blood. So the distribution of the red blood cells between the two channels here, it's like the blood. So if I have 20% of blood flow here, I will have 20% of the red blood cells flowing through here. But as you go down in size and you become like a little bit bigger than the red blood cell size or a little bit smaller, this doesn't apply anymore. What you have is, so let's say that we have, in this case here, 20% of blood flow from the parent vessel goes into this vessel. You only get like 10% of red blood cells in this. And therefore, here will be 80% of the blood flow, and this will get about 90% of red blood cells. So this is one of the causes, we think, of this heterogeneity of red blood cells throughout the network. And what we saw was like, okay, let's analyze in our channels, do this bifurcation actually apply this law? And the previous PhD before me showed that yes and no. So some channel did. So like this is would be this first channel, and this first bifurcation led to a normal, we call it classical partitioning that we expect from this Zweifach effect. But some other channel did not. And he analyzed the distribution of the red blood cells in the channel here. So where were the red blood cells before the bifurcation, to see if this had an influence on the, like, does this go to the normal partitioning, so Zweifach effect, or is it exactly opposite? And what he found is that some distribution, so like this very standard, so like cell everywhere, just at the edges, no cells because there is no space, will lead to normal partitioning. But in the case that you have such a distribution like this one, which is skewed to one side of the channel, this led to the opposite of what we expected. So this would be this case where you have a higher flow in one channel, but you don't get many cells in that channel. So we had this result and we were asking, okay, can we check like single red blood cells, what happened? Is there something there that leads to this? So what we did is we just concentrated on one bifurcation. This is an example of a video that we was taken in our lab, and I already marked 2 red blood cells because those 2 red blood cells behave differently. Let me start it again. So you can see that the orange one arrives at the bifurcation, stays at the apex here for a prolonged period of time, and then flows in one of the 2 channels. And those cells are actually called lingering red blood cells, which are not only seen in our experiment, they were shown to be present also in vivo and in silico. And there are just cells that stay stuck in the bifurcation for prolonged period of time. So what we did is we tracked all the cells that we had in our video and calculated a relative residence time, which is just a parameter for us to say, okay, like how long do they stay in the intersection, and could we call these cell lingering red blood cells? And for us, we used a cutoff like of 2. So if the cells were longer than 2 times this reference time in the intersection, we call them lingering. And from here, we analyzed different properties of the cells, but because of time, I will just explain you or show you one, which is the position, which we saw before. It's important. So we analyzed the position of those red blood cells inside the parent channel, so before the intersection. And here it's plotted, so position against, so in blue, we have the cells that we call non-linear red blood cells, and in red, linear red blood cells. And what we observe is like linear red blood cells come from the center of the parent vessel, which makes sense, because if they come from here and they need to go to the apex and linger there for a period of time, it's really difficult that they move from this side to the center and then linger. So we saw, okay, linear red blood cells in the parent vessel, we can say they come from the center. Also an interesting thing, if we put the distribution lingering and non-lingering together in the parent vessel, we have this standard distribution where in the center, they are more centered generally. And this, from the previous research, show that leads to normal partitioning, as what exactly we saw then in the distribution. The interesting thing was in the daughter vessel. What we saw is that the lingering red blood cells, so this is the daughter vessel, this is where they were lingering for a period of time, they actually stay against the wall for a prolonged period of time. We didn't expect this. So the non-lingering red blood cells, we can see they are shifted a little bit because still it's a new vessel, but they are still mostly centered, whereas the lingering red blood cells, they are really skewed against this outer wall, and they stay there also for a prolonged period of time. So if you take the total distribution of this red blood cells in the daughter vessel, it's skewed against the outer wall, which is exactly what we saw before. So our question was, can we analyze how these lingering red blood cells have an influence on the skewness? So what we did is we did a linear density lateral distribution function, both for the non-linear red blood cells and the linear red blood cells, and we added together with a weighting factor. And what we tried is to do the difference to all the previous extracted distributions and tried to minimize it to find this minimal weighting factor that could lead this distribution to get to such, for example, such a distribution. And if we plot this minimal weighting factor divided in the function that led to normal partitioning and the function that led to reverse partitioning, we can see that there is a definitely difference between the two. So to get a distribution of red blood cells that led to a classical partitioning, you cannot get there with a higher amount of lingering red blood cells in the population of the whole red blood cells. Whereas to get the reverse partitioning distribution, you need a high amount of lingering red blood cells in your whole population of red blood cells. So this gave us like a hint of one of the possibility or one of the reasoning that we could have this shifted or skewed distribution of red blood cells before a bifurcation, leading to this reverse partitioning. So this is just the conclusion. Of course, I could not show everything that we looked at. So what we saw is that red blood cells come from the center of the parent vessel for the lingering one and flow along the outer wall in the daughter vessel. They also flow slower. I didn't show this. And this will therefore influence the next this bifurcation distribution, so the partitioning at the following bifurcation. Because there were some researchers that show that lingering red blood cells influence the bifurcation where they linger. We show that they also can, or they will influence the next bifurcation. And yeah, in our case, we didn't see any influences on the bifurcation itself. but this is probably because our channels are a little bit bigger than the other researchers. And yeah, I think that's what I will, of course, acknowledge it, very important. Of course, my professor and Dr. Obrist, and so Alberto Mattegazza was the previous PhD in my lab, where I collaborated for the last results that I show. And of course, all the group which I work with, which are very, very nice to me and help me. And yeah, I would like to thank you for your attention. And yeah, I could not show everything because of time, but this was just a snippet of our research to get you interested. Yeah.
Thank you, Aurelia, so much for your talk. It was really, really nice to see something at this very small scale that is so interesting and so important in our day-to-day life, basically, because
Yes.
it governs how we live, basically. So it's really, really important for sure. So I'm asking anybody connected who wants to ask something to Aurelia to write everything in the chat so I can read the questions out loud. And in the meanwhile, if we have some questions from the studio, maybe, or maybe I can just start with a couple of questions of my own. So the first is more curiosity, and it's like, how do you design the geometry of your chips, the microfluidics chip you're using? Like, do they resemble somehow the shape these channels have in the brain, or I don't know?
I mean, of course, I think this was. No, I cannot. Okay, I mean, you can see it here. Of course, this is what we have, I think, here. This is very, very idealized. You will not have such a nice honeycomb network in your brain. But we needed to start somewhere. So that's why we decided, okay, let's start with something that we can know, okay, this is first doable. This thing is, our channels are rectangular because it's at the moment still not possible to produce such a big channel with round channels this small. So we decided, okay, we do soft lithography, we can do small channels, but unfortunately they are still rectangular. And yeah, and then the idea was to start and test. I mean, this is also something that I'm working, the project that I could not show, where we change like 1 diameters of this vessel and we change it dynamically to see the influence on the distribution along the whole channel. And if this is not symmetric, you cannot, at least for starting, you need something that you can say, okay, this is standard, like standardized, and I can draw some conclusion, and then maybe in the future try some more realistic one. But yeah, that's the idea. And the size is just a little bit bigger than the red blood cells, because otherwise we had problems with a lot of red blood cell get stuck, and then you can throw all the chip away. And since one experiment is already 1 week, you don't want to throw every week away. So yeah, that's how we came to this.
Sure. Yeah, and then my second question is actually related to the first one, and is how do you fabricate these chips? So you were mentioning soft lithography, so
exactly. So at the moment we are still doing it with soft lithography. So what we do is we did like the inverse mold with just soft lithography. So we just did with the SU8 on top of it, so the light sensitive one. And this was exactly 8 micrometer height, and then we pour PDMS on it, and then we peel PDMS off, and we put a flat PDMS layer and plasma bond them together.
okay.
I think
and
that's
you thought maybe yeah, that's the standard with microfluidics, right? More or less.
Yes.
And have you thought maybe just, I don't know, if it is possible, like to go for some 3D printing or something? I don't know if it is possible at this
I actually tried. That was the beginning of my PhD. We tried with nanoscribe to do round channels, and I managed to do round channels. The problem was the stitching between, because it's such a big I want a big channel, a big chip. And nanoscribe is really good if you need really, really small channels or small stuff. But if the whole system is big, we had problem with the stitching. And at the end, I was like, okay, my PhD is not on making new chips, so I needed to decide where to go on, and therefore we let that on the side. We are now trying with milling to do new channels to see if they are also more because the mask with time, with soft lithography with time, they lose a little bit their condition and you cannot wash them properly because otherwise you get rid of the SU8. So we are now in collaboration with another university trying to do it with milling in glass to see if we get the mold a little bit sturdier. Yeah.
Okay. Thank you very much. Really, really cool.
Ask a quick question.
Yes.
The first question is related to the advection velocity. Do we have any limitation, or do we know the range of the advection velocity in the veins, and what is the range where we have movement of blood cells within the veins?
So in the veins.
Yeah.
On the veins.
Or in your system.
In the capillary system, you normally, what we try is that we have at the inlet velocity around 1 millimeter per second.
Okay.
And then it goes down as you go more smaller and smaller, and then it comes back up when you go to the channels. Yeah.
Okay. The second question is about this lingering blood cells. We have similar phenomena when we have transport in porous media, and we have hindering objects that hinders transport of particles in the porous media, or when we have transport of bacteria or things like that. Do you see any similarity to other porous media applications when it comes to the lingering blood cells?
I think it's not my field, but I think it could be interesting. Also, this effect that I was speaking about, the Zweifung effect. I never heard it outside our community. I was an Interpor last year, and but I think this is something that could be looked at for exactly how do the transport of materials in porous media actually divide between bifurcation. I never heard if someone checked that is also following this or not. Should be interesting. I think it's something to look at for sure. And for sure, I don't know, depends on how the particle are, because there were some studies that depends on the particle. Also, if they are more stiff or less stiff, they may linger less or more. So depending on the material that you're looking at, that it's transported, maybe get stuck or get stuck more, and therefore have an influence on the whole transport in porous media. Yeah.
Excellent. Thank you very much.
You're welcome.
Thank you. Thank you so much, Aurelia, again. And yeah, there are no more questions, I guess. So I think we can close here for today's session. So let me just, yes. Okay, so yes, with this, we end today's session. And just before closing, I want to thank again all of our speakers for today, and all of the people from the PMTTT who joined me today here in the studio, and all of the team for the organization. So our next talk is scheduled for the 13th of February, so don't miss it. We will have two speakers. The first one is Negar Savar. I hope my pronunciation is not too bad. She's from McMaster University in Canada, and she will talk about precipitation flow in a confined geometry with reference to mixing, fingering, and deposition. Then we will have Milad Naderloo, who will talk about experimental and numerical investigation of sandstone deformation under cycling loading that are relevant for underground energy storage. So yeah, in case you are in, here is a slide with all the people from the PMTT teams, and in case you're interested to present to these events, just send an email to us at this contact that you see here. So with this, I end today's session, and again, I thank you all for joining. So see you next time. Bye. Bye-bye.